Well, thank you very much, Peter, for that overly generous remark and review of my work, which I'll now flesh out for the audience. It's a pleasure to be here. This is my first visit to Hungary, and I was delighted to bring my partner and colleague, Professor Sabiha Merchant, along, and we are grateful to Peter and to Andras Vara for their generous hosting and showing us some of the cultural highlights of Budapest. I'm very impressed with the National Academy of Science Education and the opportunity that has been presented to the young budding scholars here in Hungary. I wish that were available to me when I was growing up in Southern California to begin my interest in science. But what was available at the time, now over 60 years ago, was a national investment in science education in the U.S. as a result of the launch of the Russian Sputnik missile, which precipitated a major change in science investment and science education in the United States, and at the same time elicited a tremendous investment by the state of California in launching a number of new campuses of the University of California. I was the beneficiary of the Sputnik era and of the investment in public education in California. And I had the benefit, as you heard from Peter, of a wonderful high school biology teacher who took me under wing even before I started high school, as he visited one of the science fair projects, which was my most important introduction to science back beginning in the early 1960s. So I first fell in love with microorganisms when I had a toy microscope, and I took a trip to a local riverbed near my home in Southern California and collected a jar of dirty water and pond scum. I brought this back to my bedroom and put a drop of the scum on a glass slide and squinted through the plastic lens of this microscope to discover the world of microorganisms, particularly of unicellular and multicellular organisms swimming and crawling across my screen. This was simply captivating to me in a way that I really hadn't experienced to that point. I was maybe 12 years old. One evening at dinner, I was expressing my enthusiasm for this, but my father was rather skeptical that I could see anything in this toy microscope. And I remember taking offense at his remarks, and so I decided to save my money, as Peter mentioned. I saved it up from the earnings that I made mowing the lawn and babysitting for neighbors. I kept it in an envelope in my bedroom, but my mother kept borrowing the money to buy things like groceries. I became so upset that one Saturday morning after I finished mowing a neighbor's lawn, I rode my bicycle to the local police station, and I told the duty officer that I was running away from home because my mother was stealing my money and I couldn't get my microscope. They called my father in. He spoke behind a closed door to the captain of the local police precinct. He came out looking rather angry at me, but that afternoon we rode to a secondhand store nearby, and I purchased for the princely sum of $100 this student professional microscope, which you see here in my high school, one of my high school science projects, which became my pride and joy through the rest of my years in high school. But that got put away. I went to college. I went to UCLA. They had better microscopes there, and I more or less forgot about that toy microscope in this episode for many years. At UCLA and subsequently as a graduate student with Arthur Kornberg at Stanford University, I learned the tools of genetics and molecular biology and biochemistry to discover mechanisms, biochemical mechanisms of complex processes in cells, first in bacteria, then as a postdoctoral fellow in cultured mammalian cells and other more complex cells. And I decided for my independent career at the University of California, Berkeley, to begin an investigation using the tools of molecular biology to explore the mechanisms of membrane assembly. So let me just summarize what many of you will be familiar with, which is the structure of a biological membrane, and what we knew at the time that I began my career about how these membranes were put together in mammalian cells. So here's the structure of a biological membrane. This is a depiction of the fluid mosaic model, which was described first by my postdoctoral mentor, S.J. Singer at the University of California, San Diego. Bilayers of phospholipids are organized in all membranes in cells with polar head groups facing the cell exterior and the cytoplasmic face of the plasma membrane, for instance, and then acyl chains, apolar acyl chains, covalently attached to these head groups that create the insulating environment of the interior of a bilayer. Now, these bilayers would be impermeable to small molecules, charged species, were it not for the embedment of integral membrane proteins that span the bilayer, serving as selective pores, permeases, or as receptors, such as for insulin, that bind to the cell exterior and elicit a change in metabolism in the cell interior. This is the typical double-track membrane appearance of a bilayer on the surface of cells and also surrounding all of the various membranes within a cell. Now, as I think all of you know, much of the pioneering work in understanding the intracellular organelles in mammalian cells and their means of communication through processes of protein secretion were discovered by the pioneering efforts of George Palade, who is really the founding father of modern membrane cell biology. Here he is seated next to his favorite instrument, the electron microscope. The microscope had been invented by German engineers in the 1920s for inspection of physical materials that would resist bombardment and would reflect electrons propelled down a chamber shown here onto a sample specimen held in a vacuum tube shown below. And of course, such bombardment by electrons propelled by this instrument would be destructive to tissues and cells, and what Palade and his colleagues perfected over decades was the procedures to treat tissues so as to chemically fix proteins and nucleic acids and sugars so that they would then be resist extraction of water, replacement with organic solvents, very tough chemical stains to highlight particles in cells, and finally infusion with a plastic resin, allowing a sample to be processed by thin section microscopy to visualize the interior structure of a cell. Now, when Palade pioneered these techniques, the images that he produced were quite spectacular. Here's, for example, one of the favorite cells that he and others inspected. This is the beta cell of the pancreas. Palade's work largely was restricted to discovery of the structures of other exocrine cells in the pancreas that you've already heard about. And what he was able to realize was that there is an elaborate organization apparatus of membranes that encapsulate those proteins that are destined to be secreted from cells. Now, as you know, all cells that manufacture proteins make all those proteins inside the cell, in the cytoplasm. They're synthesized on ribosomes in the cytoplasm. But all cells also export by secretion some fraction of those proteins. And at the time that Palade began his investigation in the 1950s, it wasn't even known how proteins are made at all. He was the first to visualize ribosomes in the cytoplasm, and he was also the first to demonstrate that proteins that are to be secreted from cells are organized on the cytoplasmic face of the endoplasmic reticulum. He predicted that these proteins, once made, enter through a channel in the ER membrane, a special protein-conducting channel that was not known in any molecular detail, and that subsequently proteins would fold within the lumen of the endoplasmic reticulum. He was able to demonstrate through some brilliant experiments that allowed him to visualize the transfer of newly synthesized zymogen proteins among the stations of the secretory pathway, eventually to be secreted from the cell. Here, in the beta cell, we see a dramatic example of the accumulation, in this case, of a nearly crystalline form of insulin collected within these membrane-enclosed granules, poised at the point of secretion when cells are ready to discharge insulin after a meal is taken in for digestion. On a closer inspection, he and others showed that at the moment prior to the secretion of insulin molecules, these granules migrate to the cell perimeter, and the membrane surrounding the granule touches the inner cytoplasmic surface of the plasma membrane, and at a key moment, a membrane fusion reaction causes the granule membrane to fuse with the plasma membrane of the cell, resulting in the ejection of at least an initially crystalline form of insulin, but which then dissolves and is distributed into the bloodstream. Now, this membrane fusion event is a common event at many stages in the traffic of proteins in the secretory pathway. Indeed, it is a common event in virtually all the cells in our body, save for the mature red blood cell. And as I'm sure you're aware, it is particularly important in nerve cells, those nerve cells that innervate muscle cells, but also in the brain, the many different connections between nerve cells that allow nerve cells to communicate with one another. That form of a communication is intimately connected to the transmission of chemical neurotransmitters by a similar membrane fusion event that is elicited in a very time-controlled, and site-specific manner, in this case to allow the secretion of chemical neurotransmitters into the synapse, the cleft between, in this instance, a nerve cell and a muscle cell, or in the trillions of different nerve connections that mediate thought processes and emotion in the brain. Now, in 1974, when George Palade won his Nobel Prize, I had just moved from my PhD work at Stanford to become a postdoctoral fellow at the University of California, San Diego. And that very October, when he received his Nobel, he flew from Stockholm to San Diego to deliver a lecture to the annual meeting of the American Society for Cell Biology. The key discovery that resulted in his Nobel Prize is shown in this cartoon, which shows the pathway, the secretory pathway, that Palade was able to describe through a combination of morphologic, cytologic, and membrane fractionation studies, in which protein molecules must pass as they are being made from the cytoplasm into the lumen of the ER to be captured in transport vesicles that mediate traffic through a structure called the Golgi apparatus, which had been described decades earlier by a 19th century cytologist, Camillo Golgi, but which existed kind of in limbo for many years until Palade and his colleagues were able to show that this was a station in the passage of protein secreted from being trafficked from the endoplasmic reticulum. We know from Palade's work that these mature forms of insulin are processed, packaged into granules, and then discharged at the cell surface. Now, the lecture was enormously influential in my career choice, because what was most dramatic was the complexity of this pathway, the beauty of the movements of these proteins and organelles within a cell, but the complete absence of any mechanistic understanding of how this process was organized or executed. In 1974, when he won his Nobel, not a single protein molecule or lipid had been ascribed a function at any stage in this process. We knew nothing about the channel that he predicted to exist in the ER membrane through which secretory proteins would be passed as they are made. We knew nothing about the means by which vesicles could be formed to carry specific cargo molecules, to be conveyed within the cell by traffic to unique targets such as the Golgi apparatus or the cell surface. And we knew nothing about the mechanism of membrane fusion that ultimately produced the final step in the process of, for instance, of insulin secretion. I was not only motivated by this kind of description and the desire to understand the mechanism. Having been trained as a reductionist biochemist, I felt that a molecular approach would be necessary. But I was also aware from my own studies as a graduate student of the importance of understanding biological processes through functional analysis using traditional approaches that involve isolating mutations, using genetics. At the time, yeast had become a very popular model organism to study fundamental eukaryotic processes such as cell division. The pioneering yeast geneticist Leland Hartwell at the University of Washington had already succeeded in dissecting the stages in the division cycle of yeast using mutations to define the cell division cycle, or CDC genes. Thus, I thought that this process of protein secretion might also be approached initially by using the genetic tools available with a simple eukaryotic organism, yeast, to define the genetic complexity and to identify the specific genes and thus proteins that are required at each of the stages in the secretory pathway. So just a word about yeast and why it's such a valuable organism to study for eukaryotic processes. This is a scanning EM image of a budding yeast cell, Saccharomyces cerevisiae, cells that are growing in a nutrient broth under rich conditions of growth begin the cell division cycle by producing a small bud that emerges on the surface of a mother cell, and this bud continues to grow in size during the 90 minutes or so that a yeast cell takes to divide. After that period of 90 minutes, the cell develops a septum to separate the daughter cell and the mother cell. You can even see previous birth events on the surface of a mother cell by counting scars from prior budding events. These two cells then separate by a fission process, and they continue to produce more daughter cells by budding. So they can grow logarithmically over a very long period of time, producing billions of identical copies. Now, yeast cells can grow in two different life cycles. They can grow in a haploid life cycle where each gene is represented singly in the chromosome, or they can grow in a diploid cell lifestyle where two copies of each gene are available. This presents the cell with unique opportunities for genetic analysis. By using a haploid cell, you can expose the cells to chemicals that cause mutations and isolate clones by growing cells on a Petri plate and then investigate the functional loss of particular genes in mutant clones. You can then map the genes by using the diploid life cycle and produce a physical map of the genome, which was done over the years before my work began. So when I arrived in 1976 as an assistant professor at UC Berkeley, I began to explore how yeast cells might be exploited to study this pathway that Palade had described. An image of a thin section through a yeast cell, shown in my next slide, produces an impression that's obviously very different than a pancreatic beta cell. The cytoplasm of the yeast is densely packed with ribosomes. It is busy making all the proteins that a yeast cell needs to grow and divide. Yeast cells have about 6,000 genes. It's a little simpler than a human cell. But they do have some organelles that are characteristic of eukaryotic cells. For instance, they have a structure called the vacuole, which is the yeast equivalent of a lysosome. There are elements, tubules of endoplasmic reticulum that can often be seen abutting the inner surface of the plasma membrane. In other sections through the cell, you can see a double membrane surrounding the nucleus. You can see mitochondria, and you can even see occasional elements of what appears to be a Golgi apparatus. My attention, the attention of my budding lab in 1976, was drawn to the appearance of a small cluster of vesicles underneath the growing portion of a daughter cell. These vesicles were known to carry enzymes that are secreted into the cell wall surrounding the plasma membrane, where they serve to allow yeast cells to harvest macromolecules from the environment to provide sugars and amino acids for growth. We imagined that in addition to a role in secretion, these vesicles would carry a bilayer, which would include, of course, integral membrane proteins that would become the building blocks for the growth of the plasma membrane of the cell. Thus, one of my first graduate students and I, my first graduate student being this young fellow here, Peter Novick, shown pipetting at his bench in my lab in 1978. Note the long hair. We all had long hair back then. I too had long hair, but I have much less hair at this point. Peter was a brilliant graduate student, and he and I systematically began an effort to identify mutations that would affect the process of secretion. Now, the prediction made from the simple hypothesis that I posed in the previous slide is that any vesicle required for secretion would also be required for the assembly of the plasma membrane. And thus, genes required for the production, migration, and fusion of such vesicles would be essential genes. That is, if you deleted such genes, the cell would die. And having a dead cell isn't very good. You need a cell that you can work with. So fortunately, over many decades, microbiologists had devised techniques to evaluate functions of essential genes. You can identify such essential genes in a simple organism such as yeast by exposing the cells to a chemical mutagen, which will occasionally cause mutations in single amino acid residues that control the stability of the folded state of the protein molecule. Some mutations will allow a protein, for instance, to fold normally and function at room temperature, while causing the protein to unfold at a slightly higher temperature. Yeast cells can grow over a range of temperatures from very low up to, say, 40 degrees centigrade. And so a convenient range that one uses to examine the function of a protein with a so-called conditional mutation is something from room temperature up to 37 degrees. So what Peter and I did was to begin by simply exposing yeast cells to a chemical mutagen. Cells are plated on a Petri plate, and by very simple techniques, it's possible to identify colonies that will grow at room temperature, but not at 37 degrees. Now, of the 6,000 genes in the yeast genome, about 1,500 are essential genes. So it would be conceivable that if you were to conduct such an experiment, you could find mutations in 1,500 genes. But you want only those mutations for this investigation to be those that affect the process of secretion and not other cellular events such as protein synthesis or nucleic acid synthesis. So what Peter devised were very simple techniques that allowed us to look for cells that, when warmed to 37 degrees, cause the enzymes that would otherwise be secreted into the cell wall to build up inside the cell without being exposed to the cell exterior. Now, in 1978, near the beginning of 1978, Peter had identified the first such mutation called SEC1, short for secretion 1, which had these properties. It was temperature sensitive for growth, and it caused two different secretory enzymes to build up inside the cell. At just about this time, George Palade visited UC Berkeley for a series of honorific lectures, and I had the pleasure of introducing him to our work on protein secretion. But more importantly, the graduate students organized a dinner in his honor that evening after his first lecture, where Peter told Dr. Palade about the initial characteristics of this secretion mutant. Palade said, well, now, of course, you must inspect the cell by thin section electron microscopy to see where the enzymes that were building up inside the cell accumulated. We had anticipated, of course, doing that experiment, rushed ahead to do it as quickly as possible. And by the summer, one morning, Peter called me excitedly down to the basement of our building to examine the first such mutation, comparing cells grown at the permissive growth temperature and at the restrictive growth temperature. And one of the great moments of my scientific career came when we had a look at the behavior of that first mutant. So recall from a slide a moment ago the small number of vesicles that accumulate within the bud portion of a dividing yeast cell under normal circumstances. In contrast, in this mutant, and in many more isolated subsequently, we could show that the vesicles failed in the last step, failing to fuse with the plasma membrane, and instead secretory vesicles, mature vesicles, accumulated throughout the cytoplasm. Thousands of these vesicles accumulated over the course of a couple of hours of incubation at 37 degrees. Now, as these cells were dying, if the cells were restored to room temperature, these vesicles re-engaged the proper process, secretory enzymes were produced, and new vesicles could be made to continue this process, and the cell would resume growth. Well, this was, of course, a very exciting observation, and led over the next years to the isolation of many more such mutants, initially in my lab and in the lab of other former students of mine in their own laboratories. And so now let me rush forward some years to show you a pathway that we were able to devise by using a combination of genetic and cytologic techniques, a pathway that looks essentially identical to that developed by Dr. Pallotti using a very different approach. We could show that in yeast, proteins progress from the endoplasmic reticulum in a vesicle intermediate to the Golgi. Postdoctoral fellows Tom Stevens and Scott Emmer in their own labs subsequently were able to isolate a number of genes that are required for the sorting of proteins in the Golgi apparatus, some of which are secreted and others of which are diverted to a structure that is the yeast lysosome, so a very large number of genes. And finally, those genes such as SEC1 that are required to convey proteins from the Golgi apparatus to the cell surface. So although this was formally a replication of what Pallotti had done, what we had was the bonus of a great deal of molecular information concerning genes that are required for a bidirectional flow of vesicles and those that are required for targeting of vesicles to the cell surface. Subsequently, another brilliant graduate student, Ray Deshay, in my lab, used a different approach to identify the genes that encode the channel protein in the ER membrane that Pallotti had speculated on required for the translocation of proteins as they are made across the bilayer. Now, at this time when we had this information, it wasn't yet known what these genes encoded. It became possible within a few years to be able to molecularly clone these genes and to discover at least the predicted protein sequence of the genes. But at the time that we did this, for many of the genes, there were no homologs yet in a database because the other genomes had not yet been sequenced. However, eventually, the human genome was sequenced, and it became clear that yeast and humans are very similar in this, and certainly in other pathways. And I use this comparison between a drawing of Charles Darwin and a cluster of yeast cells to establish what is now a universal property, and that is the conservation of function over 2 billion years between the origin of yeast cells and the emergence of the human race. The machinery within a cell required for this and many other pathways is fundamentally conserved. Indeed, the first gene that we isolated, SEC1, has its equivalent in the brain that's required for the docking and fusion of synaptic vesicles at the presynaptic membrane. This conservation of function became an obvious target of opportunity for the biotechnology industry, which was then growing up in the San Francisco Bay Area. And I had the opportunity to help a local biotech company by the name of Chiron to introduce different human or viral genes into yeast to use yeast as a platform for the production of commercially viable quantities of such proteins as human insulin. And now, as Peter mentioned in his introduction, a company in the Netherlands, Novo Nordisk, is responsible for manufacturing 1/3 of the world's supply of human recombinant insulin, which is made in giant fermentation vats, growing up yeast cultures that prolifically secrete mature insulin into the growth medium. Chiron also introduced the gene for the hepatitis B surface antigen into yeast, a membrane protein, and that surface antigen accumulates within transfected yeast cells in little vesicles that can be isolated from broken yeast cell preparations. And for any of you who've been vaccinated against hepatitis B, you have received little yeast vesicles that contain this hepatitis surface antigen. Well, this was all well and good. The knowledge that these pathways are conserved was comforting, but having been trained as a reductionist biochemist, I really wasn't satisfied with that level of insight. I wanted to explore the function of the gene products. Having their sequence showing they are similar to those found in the human genome doesn't necessarily tell you what the proteins do, and for that we felt it would be essential to have a biochemical approach similar to what had already been developed by Jim Rothman in his lab, initially at Stanford and eventually at Yale University. So I will now tell you about a story that we use to try to understand how vesicles get made using a biochemical approach, and I'll tell you how this has informed at least one rare human craniofacial disorder. So in the course of our investigation, another talented postdoctoral fellow in the lab, Chris Kaiser, was able to show using genetic and cytologic inspection of mutant yeast cells, that a vesicle intermediate conveys proteins from the endoplasmic reticulum to the Golgi. He was able to show that a subset of the SEC genes required for that limb of the pathway are involved in the formation of vesicles that bud from the endoplasmic reticulum, and another set of proteins that are required for the fusion of those vesicles at the Golgi apparatus. In cloning and sequencing these genes in yeast, and a corresponding effort in Jim Rothman's lab to clone and sequence two proteins that he had described to be involved in vesicle fusion at the Golgi apparatus, we discovered that the sequence homology had a functional equivalent in the fusion reaction. Two genes, yeast Sec17 and Sec18, are equivalent to two proteins that Rothman had purified from brain extract called NSF and alpha-SNAP, and they function exactly at the predicted stage in membrane fusion. We were interested in those genes that are required to produce vesicles by a budding reaction from the plasma membrane. And again, I had the good fortune of two brilliant graduate students, David Baker, began by investigating a means of breaking open yeast cells that would allow the part of the secretory pathway to be preserved with isolated membranes and soluble cytoplasmic proteins. We were able to show that in the lysate that Baker had produced, if the ER membranes are protected by a gentle lysis procedure, they remain very large and will sediment very rapidly when an extract is suspended and then centrifuged at low speed. All of the ER membranes would pellet to the bottom of the tube. However, if these large ER membranes are incubated with crude yeast cytosolic proteins and ATP during the course of approximately 20-minute incubation, small vesicles are formed in the test tube that cannot be centrifuged at a low speed and instead remain in the supernatant fraction, requiring a higher speed for their capture by pelleting in an ultracentrifuge tube. This became the assay, the vesicle budding assay that we used, and we were able to show, very importantly, that the formation of these small vesicles depended upon the SEC proteins that we had described to be involved in vivo in the formation of these vesicles in living cells. So the very same genes are required both in cells and in vitro, and this then reinforced our knowledge and ability to use this information to purify functional forms of the proteins required for the vesicle budding reaction. Eventually, with pure proteins achieved over the course of several years with much effort by many students, culminating with some very important work done by Charles Barlow, a postdoctoral fellow in the lab, we were able to discover that the proteins required for the budding reaction comprise a complex set of coat proteins that envelop, surround, and pinch vesicles from the endoplasmic reticulum. Here is one such experiment that we conducted in collaboration with a brilliant morphologist in Geneva, Leolia Orci, when Barlow took the pure set of proteins, mixed them with ER membranes, formed vesicles, and sedimented these vesicles to the bottom of an ultracentrifuge tube. Samples processed for microscopy were shipped to Geneva and inspected by Orci, and we observed a very uniform population of these transport vesicles, coated evenly with the very proteins required for the formation of these vesicles in both in vivo and in the cell extract. Now, this coat, at least at this level of inspection, looked very similar to a molecularly distinct coat that Jim Rothman had discovered to be required for traffic of vesicle-mediated traffic within the Golgi apparatus. And since his discovery was first, I thought it would be appropriate to call his coat COP1 and the coat that we discovered subsequently to be called COP2. Over a long period of time and work in many other laboratories using structural approaches, we now know with atomic resolution exactly how these proteins form a coat and how they pinch a vesicle from the ER membrane, and I'll summarize this in this slide. The coat consists of two layers of protein, sec protein products. An inner layer consisting of one GTP binding protein and a heterodimer of two gene products, Sec23 and Sec24, bind to the cytoplasmic surface of the ER membrane, and through a direct interaction with this subunit, Sec24, proteins that project a sorting signal from the membrane to the cytoplasm, a signal that is required for their traffic from the ER to the Golgi. A signal is recognized by one of several different binding pockets on this Sec24 molecule. They are then bound non-covalently, captured, held in place, and then surrounded by two other Sec proteins that form a cube octahedral polyhedral structure that forms around the membrane, conforms it to about 100 nanometer particle, and then by completion of polymerization pinches the vesicle from the ER membrane, severing it by fission from its ER membrane. The coat subunits are then shed, and the vesicle, a naked vesicle, is revealed that is directed by membrane fusion apparatus to the Golgi apparatus, where cargo molecules are then transferred for the next stages in the pathway. Now, in the course of these experiments, which were done initially entirely on yeast cells, we decided for various reasons to explore this pathway in mammalian cells using a combination of morphologic and biochemical techniques. And as we were establishing this budding reaction from mammalian membranes, I was approached by a clinical geneticist from, at that time, Johns Hopkins University, Simeon Boydjev, who had, in collaboration with a Saudi investigator in Riyadh, discovered or investigated a family, a Bedouin family with a rare craniofacial disorder called CLSD that has a quite unusual effect in cranial morphology. But most importantly, this family has a mutation in an invariant residue of one of the four paralogs of the, one of the two paralogs of the Sec23 gene, the subunit of the inner layer of the COP2 coat. Children homozygous for this CLD mutation in one of the Sec23 paralogs show this kind of facial morphology. They have failure of complete bone closing. The skull surface remains open. The soft spot on the top of their head never closes. They develop cataracts. They have brittle bones. But at least as far as one can see, they're still actually fairly normal at this stage in development, and they may still be alive, I'm not sure. Through this collaboration with Simeon Boydjev, we obtained tissue samples of the kids, the homozygous kids, mutant for this copy of SEC23, one of the heterozygous parent carriers who have no obvious pathology, and compared by thin section analysis the homozygous, the heterozygous, to a normal skin fibroblast by thin section analysis, again conducted in collaboration with Lelio Orci in Geneva. And the image comparison is quite striking. The top panel is a thin section through a normal fibroblast, showing a fairly smooth endoplasmic reticulum with some granular material in the lumen of the organelle. One of the parents, a carrier with no evident pathology, at least evident in the patient, showed nonetheless some distortion of the ER membrane with, again, some accumulated material in the lumen. But at least one of the children, a homozygote, the fibroblasts collected from this child show quite an enormous distortion, tubular, bloated distortion of the endoplasmic reticulum with a great deal of accumulated material. We've been able, over subsequent years, to understand the exact nature of this mutation, how it interferes with the capture of molecules like procollagen, which are actively secreted by skin cells. Nonetheless, it's surprising that these cells survive and grow and that the patients grow when the very same mutation in the yeast gene is lethal to a yeast cell. The reason that these cells from the patients survive and that the patients themselves seem to flourish is that there are two copies of the SEC23 gene. It just so happens that one of them has a selective effect on these cells, the fibroblasts. Well, so I've taken you through a long journey from microbiology to genetics to morphologic inspection and finally to a biochemical analysis. And I would have been content to continue with my experimental life, which was interrupted by a call from Stockholm now 10 years ago. At the moment that Tom Südhof and I shared on the stage in Stockholm, I was delighted to be recognized by the King of Sweden. But something even more interesting happened. After I received the call from Stockholm, several weeks later, I and my fellow laureates received an inquiry from something called the Nobel Museum in Stockholm. Every year, the Nobel Museum asks new laureates to send in some artifact that reflects the development of their interest in science. I didn't think it would be particularly interesting to send in a plate of yeast cells colonies of yeast cells on a Petri plate. Instead, that microscope, that old microscope that I earned the $100 for back in 1962, was sitting collecting dust in my closet because neither of my children showed any interest in science. So I dusted the microscope off, and I sent it to Stockholm. And you can visit my microscope on display in the Nobel Museum, together with a caption in English and Swedish that tells how I had to run away from home to begin my career in science. Thank you very much for your attention.
So thank you very much for this brilliant talk, and again, congratulations on the discovery which actually not only changed the scientific world and open up new discovery ways, but also helped a lot for the patient's benefit. So, students, the stage is yours. Questions? Who has questions? I can't see.
There's a hand up there. Some young woman had a hand up. Oh, she's getting the microphone.
Thank you for the amazing presentation. It was absolutely stunning. My question would be about, you mentioned near the beginning that during the first few years of the research on this topic, you wanted to separate those cells that were impacted, their growth was impacted by temperature change, so that were ones that wouldn't grow above the temperature of 37 degrees Celsius. Could that have been modified if, for example, you changed the pH of the substance they were in, or, for example, added different salts to the substance?
Yeah, so you raise an interesting point. There are many different kinds of mutations that exert conditional effects on the growth of cells. The most convenient one to look for those genes that are essential for some aspect of cell growth is this simple temperature change. You could change the pH, you could change the salt conditions of the medium, and find other cells that are conditional in respect to the medium composition. But those would be more difficult to ascribe to essential functions for normal growth in rich broth. We looked exclusively and specifically for those mutations that confer a growth defect at 37 degrees, and which at the same time cause a defect in the secretion of two different enzymes. And that allowed us to identify initially a couple of dozen different genes, each of which exerted a very specific requirement at a different stage in the pathway. But there are other approaches for conditional growth that are used in microorganisms, and one's limited only by one's imagination and looking for different growth conditions.
Thank you very much.
Thank you. Next question. Students or anybody?
Thank you very much. That was very exciting. I was wondering whether there are any homologs of any of these molecules in prokaryotes.
In prokaryotes.
Or this is very eukaryotic.
Yeah, excellent question. I mentioned very briefly there's a GTPase called SAR1, which is involved in initiating the polymerization of the COP2 coat. There are prokaryotic homologs of SAR1. It's not clear where or how they function in cell growth in bacteria, but they exist, and there may be some intelligence on this now, but I haven't heard anything more than that. Now, some of these genes may also exist in archaeal species. There are some bacteria and archaeal species that have intracellular organelles, so it's quite possible that these molecules exist there. And of course, these molecules also exist in protozoa, since they are bona fide eukaryotes. But the machinery largely evolved after bacterial evolution and is more unique to eukaryotes than to prokaryotes.
Thank you very much.
I have an easy question. So how your relationship with your mother changed when you went to the police station and reported her action?
I don't remember my mother's reaction, but I think my father came out from the captain's office looking very severe and angry with me. But I learned a valuable lesson that afternoon because we drove from the police station to a pawn shop in a nearby city and bought the microscope. So I had my way.
Thank you.
My mother's reaction was more extreme when I started university and changed my mind. I had initially entered university with the idea that I would go on to medical school and become a physician. But in my freshman year at UCLA, I had the opportunity to work in a research laboratory, which completely changed my perspective on my career. And I came home after my first year to tell my parents that I was not going to be a real doctor after all. They were quite disappointed, but they got over it.
I think finally they also were very pleased at the end of the day. Questions?
Well, I can answer questions privately during the break.
Yes. So thank you very much for everybody. Thank you for the speakers. Randy. Randy, please don't go, because now I would like to ask the chairman of the curatorial board to deliver the foundation prize to you.